Our topic today is about genomic medicine and its impact on public health. Our presenter is the influential Dr. Alfaisal Alghalyini, who established the developmental genetics lab that later evolved into the Center for Genomic Medicine at King Faisal Specialist Hospital. He is an authority in the area of Mendelian genetics with more than 480 published manuscripts that describe his lab's discovery of hundreds of novel novel disease genes in humans. He is frequently invited as a speaker at local, regional, and international conferences on the editorial board of prominent human genetic journals, and the recipient of numerous prestigious awards, including the William King Bose Award in Medical Genetics, King Salman Award for Disability Research, and the Kurt Stern Award. Please put your hands together to welcome Dr. Alghalyini. This is the
advanced slides and the laser pointer.
Yeah.
Perfect. Thank you so much. Assalamu alaikum. Good afternoon, everybody, and truly honored to be here. And I'm excited for two reasons. A, having given an in-person talk in 2 years, more than 2 years. So it's good to awaken that dormant part of my brain, having given that in-person engagement in so long. So let's see if we can do well today. And second is because of the topic. Genomic medicine is near and dear to my heart, and I'm hoping that I will get you hooked after you hear some of the exciting developments in the area of genomic medicine. Now, you don't need to become a geneticist. Genomics is going to infiltrate every piece and aspect of medicine. So I guarantee you, if you're one of those people who make a pledge to never come near genetics, you will never become a good doctor going forward. So this is no longer an option. You absolutely need some minimum in order to survive the avalanche of genomic data that's going to drive your field, whatever it is, even surgery, in case you were thinking. Okay, so I chose the title of a primer. I'll try to resist moving about because I was told I need to remain under the focus of the camera, but that's not my style. I like to move, so I'll try to resist. Anyway, so the title of my talk is a primer on genomic medicine. There's just no way I'm going to cover all the exciting aspects of genomic medicine in 50 minutes or so, but it's a primer. I just want to prime you to get you interested, to look up some of the stuff I'm going to be talking about, and hopefully you're going to get really excited about its potential to reshape the way you look into whatever field of medicine you'd like to focus on going forward. So let's get started. I guess the revolution in genomic medicine is really part of this larger revolution that we call the digital revolution, right? We're moving from the analog to the digital, and this has found its way into genomics as well, because genomics, if you think about it at the most basic level, it is truly digital when you think of the software of life. Our hardware parts are actually made of the software. So the DNA code think of it as the software that codes for the little robots in your cell that make up your cell that we call proteins and the like. And collectively, they make up who we are. So our hardware parts are actually encoded by the software. So if you think about it that way, since we all have different software, there's no reason to think of our hardware to be identical. Our hardware parts are not identical. And so we should not expect medicine to deal with each one of us exactly the same way across the board. Each one of us is a unique individual. And that's the whole concept behind personalized medicine, individualized medicine, whatever you want to call it, but it's all part of this genomic medicine, because you can only individualize and personalize if you understand the software that makes up your body. And so I guess before I get into all the exciting aspects of genomic medicine, I decided to do something a bit weird. So I decided to invent a time machine and jump to year 2080. And I was able to grab a book on the history of medicine from the year 2080 and I was able to step back, and I will be reading you some of the excerpts of that book. And let's see how realistic that would be, and let's see what your reaction will be like. So I'm going to start with excerpt number 1 from the introduction of that book. And it goes as follows. It behooves us, as we enjoy an unprecedented level of health in our population, to reflect back on the not too distant past and ponder the giant steps that medicine took to bring us to where we are. I know that most of the readers of this book will be too young to know most of the historical facts that I will summarize in my book. In fact, I made a point of including extensive historical documents to prove the accuracy of what I will summarize, because I know that many will find it very hard to believe that such was the state of the art in the medical establishment just a couple of generations ago. All right? So that tells you something about that book that I'm reading from. And let's take another excerpt from the chapter on reproductive health. And it reads as follows. We now take for granted that our gadgets tell us immediately when we consider marriage choices, whether there is any incompatibility in our genomes. The minute we reach the legal age of adulthood, we're given the choice to unlock parts of the genome data that was generated for us when we were babies. Once we unlock it, we will know the kinds of diseases that we are carriers for. Of course, being a carrier does not affect our health, but can have dire consequences if we marry an individual who's also a carrier. With a click of a button, we know whether a potential mate is compatible or not. It is hard to believe that this very simple step that we take for granted as we plan our family life did not exist or existed at a very small scale even after the turn of the 21st century. What may be shocking to many of you is that even the primitive test that was available as part of the so-called healthy marriage program was not conducted properly, and as many as half of those incompatibles went ahead with their marriage plans anyway. Just imagine sickle cell anemia, thalassemia, countless esoteric diseases that we barely encounter anymore in our society, except for the elderlies who were born early in the 21st century, used to plague the Saudi healthcare system. Okay, now let's move on to the fetal genome profiling. And let me read you an excerpt again from this chapter. It goes as follows. The routine we are used to now in terms of having, by the way, for the latecomers, I'm reading from a book that was written 2080 on the history of medicine. I invented a time machine, got that book, and I'm reading excerpts from that book now. The routine we are used to in terms of having fetal DNA fully sequenced from the maternal circulation at 7 weeks of gestation to know whether the fetus has any genetic diseases was nearly sci-fi just 2 generations ago. It is hard to understand why the medical establishment was so focused on Down syndrome, even though the rate of de novo mutations that can cause much more devastating disorders is actually higher. When early versions of this test became available after the first decade of the 21st century, there was an outcry that it should be limited to Down syndrome and other aneuploidies, but not expanded to scan the whole genome for any other disorder. The argument made by the opponents sounds baseless right now. Designer baby, gender selection, discrimination, breach of baby's autonomy, etc., etc. As usual, people were and will always be afraid of new technologies, but we should always try to learn from the past. Now let's move on to something near and dear to the heart of the pharmacologists here, Dr. Hatouf and others. This is an excerpt from the chapter on drugs, and it reads as follows. You've probably watched the occasional news that a malpractice lawsuit was brought against a careless doctor who prescribed a drug without ensuring that the patient's genome file was logged in the system. But did you know that most doctors, as recently as 2022, did not even study in their medical school that you respond to drugs based on your own DNA profile? Believe it or not, doctors were taught and trained that if a drug works in a majority of patients, then that's good enough to prescribe that drug to all patients with that illness. What's worse, if a drug is known to cause a side effect in a small percentage of patients, it was totally okay for the doctor to still prescribe that drug and simply tell his or her patients, we'll try our luck with this drug, and if it causes you a side effect, then well, hard luck, I've already warned you. And on the chapter on the treatment of genetic diseases, the next excerpt reads as follows. Although we have greatly minimized the burden of genetic disorders in our society, the very few with genetic disorders whose parents decide to keep their pregnancy, or those whose diagnosis is missed when their fetal genome file is generated, are immediately diagnosed as soon as they're born. I have personally interviewed old Saudi doctors who gave me heart-rending accounts of how it used to be routine for families of children with genetic diseases to go shopping between so many doctors for years and years before a diagnosis is made for their children. What is more heart-wrenching is how these children were often left without treatment even when the exact genetic lesion was known. They stood helpless in correcting the genetic defect to give these children and their parents hopes in a healthy life. What we now take for granted in terms of editing these patient genomes to cure them was very much sci-fi at the prime time of their practice. And moving on to the field of infectious diseases and microbiology, here's an excerpt from that chapter, and it goes as follows. You have a fever. Your gadget gets a microdrop of body fluid and tells your doctor within minutes that which microbe caused your fever and what drug is best suited to kill it. If you think this is ordinary, think twice. As late as 2022, the way infectious diseases were diagnosed was through an archaic method called culture, where samples are taken and grown under selective conditions to see what microbe will grow within a few days. Many times, the cause of the illness is never recovered, and doctors used to keep throwing one antimicrobial agent after another to try their luck. Even when a specific microbe was identified using this primitive method, antibiotic resistance was a global public health emergency. In fact, the situation was so dire that the President of the United States had to convene a group of experts who issued a report in September 2014 on how this emergency may be thwarted, and of course, the study of genomes of pathogens was one key recommendation. Luckily, since we learned how to edit these genomes to rid them of the components that drive antimicrobial resistance, we ushered in an era in which antibiotic resistance can no longer be viewed as a threat to the prosperity of our nation. And finally, on cancer, here's an excerpt from the chapter on cancer, and it reads as follows. War on cancer was declared by one US president in 1971, but it has taken us a very long time to reach the stage we are at today, where cancer is either a curable or at least a manageable chronic disease. Receiving a diagnosis of cancer, especially in adults, was the synonym of death sentence as recently as 2030, before personalized therapy became the standard of care. Again, it may be very hard to imagine in hindsight why it took the medical establishment so long to use the body's own immune system to combat cancer, even though it has been known for many decades that immune deficiency leads to cancer and patients with autoimmune diseases are protected from cancer. The personalized cancer vaccine that we take for granted today was considered unthinkable in the beginning, even in the beginning of the 21st century. Okay, so that's a lot to digest. Of course, I lied. I didn't invent a time machine, and this is all my predictions of what life is going to be like in 2080. But what I would like to spend the rest of this presentation is why and how I base these predictions. They're based on exciting data that are coming out of work from us and others in the field that are clearly showing us the path forward where medicine is going to be radically different from what we're taught at this point in time. So it all started with the Human Genome Project. As you guys know, the Human Genome Project was a multinational, multi-billion-dollar project that aimed to sequence the human genome to provide the blueprint of that software that I mentioned at the beginning of my talk. And what was the game changer is that although the initial Human Genome Project was meant to form that blueprint for the entire humanity, it was based on a handful of individuals. But as the sequencing cost dropped, it became possible to sequence the genome of an individual. So the first time in history, we're able to peek into the software of single individuals, and that's exactly what's needed in order to deploy all these exciting tools of genomics and personalization in healthcare. And this is all thanks to the advent of what we call next generation sequencing, which is a very disruptive way of thinking of sequencing from the way we used to sequence DNA, which was called Sanger sequencing, to this new technology that massively and in parallel sequences your entire genome in a matter of hours. And I don't know if you've heard the news, the most recent record was a sick baby in a NICU at Stanford whose genome and diagnosis was finished within 6 hours. So this just keeps getting more and more exciting. So let's take it one at a time. So this was my little plug-in for the monogenic diseases, something near and dear to my heart. I told you in that history book, imaginary history book, how monogenic diseases used to really suffer in finding a diagnosis, let alone treatment. These patients used to shop for years and years from one doctor to another. Each one of these patients, let's see if this pointer works, yep. Each one of these patients has a monogenic disease, a devastating disease caused by one single glitch in that software. And you would think, wait a second, this is a piece of cake. If it's that simple, if it can be traced to a single variant, why is it so hard to identify the variant? Well, because the blueprint wasn't there, and then we didn't have the tools to sequence the entire genome. And after the genome was already sequenced, it was very difficult to filter out what's benign and what's pathogenic. But here, we're making such great progress that I'm absolutely certain of this prediction, where every single patient with a monogenic disease will have the diagnosis done in a matter of hours. I just gave you a little example of where we're at now, that example from Stanford. We do have, for example, the Flash exome service, where we sequence the coding part of the genome of sick individuals and report back the result within 48 hours. So you can totally see that we're moving very, very rapidly to that future where, these diseases are diagnosed very, very quickly. And so just to give you an idea, and again, not to brag about it, but just to show you how these things are possible if you have the right resources, our center alone was capable of making equivalent number of novel gene discoveries as the centers of excellence for Mendelian genomics in the US combined. And in fact, this was profiled by Science a few years ago in order to highlight the power of the local population to identify novel gene involvement in the area of monogenic diseases. Okay, so that was that. And then if you remember, the other excerpt I read was on fetal genome profiling. And again, this is not me having, a senior moment, thinking of what may be happening in the future. This is actually based on what's happening now. As you know, being able to profile fetal genome from maternal circulation, so-called non-invasive prenatal testing, is something that's been talked about for a while, and it was only 2011 that a company by the name of Sequenome was able to license the technology from Professor David Liu from the Chinese University in Hong Kong, who was the winner of the King Faisal Prize in Medicine 2014, where he pioneered the profiling of fetal genome from maternal circulation in order to identify chromosomal abnormalities. And as you can imagine, this evolved very quickly into something much more competitive, where companies are competing as to how much more we could tell the parents about the fetus. So it started as aneuploidies, and then it expanded into microdeletions, and there are labs that are offering de novo testing also. So variants that are absent in both parents but present in the child as a diagnostic method. And I totally see the future where all genetic diseases will be tested as part of that genome profile. Okay, I lost something and I don't know what happened. I'm still thinking. Oh, okay. What happened? Did I press something wrong? Okay, so I was critical of the Healthy Marriage Program, if you heard the excerpt I read from that future book, and that's for a good reason. Because think about it. Each one of us is a carrier of a few deleterious variants that cause terrible, terrible monogenic diseases, such as the ones I showed you on an earlier slide. The problem is these are recessive. So if you're a carrier, you have no clue that you have these variants. They're only going to reveal themselves if you're married to someone who's also a carrier for the same gene, and then you have a child who is biallelic, who has the pathogenic variant from his mom and the pathogenic variant from his dad, and you end up having a recessive disease. So it stands to reason that we should be screening everyone to know what we're carriers for in order to make an informed decision. So if you remember what I mentioned in that excerpt, where the gadget will tell me exactly if I'm interested in marrying someone, we can immediately tell if we're compatible or incompatible with just an app on our phone, right? So that's not crazy sci-fi. It's actually gonna happen very, very soon. In fact, it's already happening by some companies. And as to, I forgot to mention, the reason I was critical of the the Healthy Marriage Program in Saudi Arabia is that it wanted to focus on thalassemia, sickle cell anemia, which is fine because when it was first launched, the technology wasn't there to expand beyond that. The problem is the program did not prepare the public sufficiently with proper counseling that we ended up having most people who are incompatible going ahead with their marriage plan anyways. It was really very depressing to see that such an expensive program ends up with people who are incompatible still deciding to go ahead with the marriage. And you sit down with these people and you hear personal accounts and they all tell you the same story. We've already made our marriage plan. It was just a last minute thing, a formality we needed to get for the marriage contract. So after we were told we were incompatible, it was very difficult for us culturally to walk out on this, marriage proposal. Now, things are changing, and I'm really impressed that the number of incompatibles who are still moving ahead with their marriage plans is dropping. But I want to show you something to inspire you. So what Australia did was really inspiring. So I co-chair the International Advisory Board on the so-called McKinsey mission. So McKinsey was a little girl who died of spinal muscular atrophy, which is an autosomal recessive disease that's lethal, although now there is actually a gene therapy for it, so it's treatable. And so they decided to turn their tragedy into something positive. And they lobbied their government because after realizing, wait a second, you mean we lost our daughter for a disease that you could have told us about before we conceived of our daughter? I mean, that's inconceivable. Why did you not tell us? So they approached the government, they lobbied the government, and the government agreed to do expanded carrier screening, preconception carrier screening, and they just concluded the pilot phase of the program. And I'm telling you, this is going to be copied by many, many countries. And of course, we should be pioneers in this because we have a very high rate of consanguinity in the country, which increases the chance of incompatibility between individuals. But this is just to give you a flavor of what the future is going to be like. These surprises of having a child with a recessive disease that you had no clue about is going to be history. And people in these history books are going to look back and think, you guys were out of your mind allowing this to happen in the first place. Okay, so again, when it comes to this futuristic kind of prediction about where your genome file being uploaded and you can't order anything on the computer system, on the EMR, unless you check for compatibility. This is already happening. So at St. Jude, they were really light years ahead of everyone else. So at St. Jude, they incorporated that into the EMR such that when you click to order a medication that is linked to a pharmacogenomic variant already uploaded in the system, it will give you that warning that, wait a second, you can't give this drug or you need to reduce the dose and so forth. And we're working as part of this project that I have with Dr. Hatouf, we're trying to understand the variants that underlie side effects of medications in the country in order for this to become a reality here as well. Okay, so when it comes to infectious diseases, I still find it mind-boggling that we have the technology where if I get a sample of whatever fluid that may be involved in your infectious disease, whatever it is, pneumonia, sepsis, I should be able not only to identify the pathogen, I should be able to give you the antimicrobial resistance profile. And yet we're still using the archaic method of culture. And again, I'm really excited that we're developing in our department a platform so we can replace that with metagenomics, being able to screen your body fluid for all pathogens and report back to you the antimicrobial resistance profile. And you can see that countries are really investing in this. This is big deal. It's not only good for patient care, but to the point we were discussing with you about community health, this is very important for public health because you can really drastically reduce antibiotic resistance when you know exactly which microbe you're treating every time, as opposed to the guesswork, which is very, very common in today's practice. And thirdly, it has to do with national security. You're probably aware of this drama that happened with the MERS, where the pathogen that underlies MERS was actually not sequenced in the country. It was shipped to the Netherlands, and then there was a little bit of a fight over who owns the data of MERS. And this could have all been prevented had this sequencing happened in-house. And again, I'm really optimistic that things are moving in the right direction there with the development of metagenomic infrastructure, as I mentioned earlier. Okay, cancer. I mean, so much is happening in cancer. It's becoming really the poster child of what genomic medicine promises. And so right now, patients get profiled in order to identify variants that could predict their response to medications. And this is one of the, landmark trials sponsored by the NIH. It's called the MATCH trial, where patients with cancer get profiled as quickly as possible. And more than 50% of these patients will end up having variants that predict their response to very specific therapy. And this is just the beginning. And as I said, we talked about the use of body fluids to monitor your health. And cancer is just a perfect example of this. Just imagine, if I know what caused your cancer, what drove your cancer, the driver variant in your cancer, and I treat your cancer, I can easily monitor you through so-called liquid biopsy, try and identify this variant at a very, very small level. And this is one of the landmark papers. And again, I'm trying to show you really landmark papers that set the stage for what the future is going to be like. So in this paper, they show the superiority of using circulating tumor DNA over regular monitoring. Because as you can imagine, these technologies are so sensitive, you should be able to identify that variant at a very, very small fraction, and that will be far, far, far before your CT scan will show a recurrence of a lump or things like that. And here's another example of using it as a biomarker study, as a biomarker. I did mention something about your immune system in that book, if you remember. And again, this is not something that's baseless. It's actually based on exciting developments in the area of immune therapy. Now we understand that cancer is very smart in evading your immune system by blocking what would be a very effective response by your immune system. And so these PD-L1 and PD-1 inhibitors that remove that break that cancer is very good in mounting, you are allowing the immune system to mount a very strong response to cancer. And if you go to any oncologist who's used these drugs, they will tell you wonder stories. It's almost like a miracle. You see the tumor melt before your own eyes because of the power of your immune system. Now, I'm not saying that every tumor could be treated this way, but this is just the beginning of showing eventually what the immune system will be like. I remember when I was a medical student, this was mentioned as a futuristic idea, and we were thinking, whoa, that would be great. But now it's happening. It's not the future anymore. It's actually reality. And we talked about the cancer vaccine, being able to tinker with your immune cells in order to make sure that you don't get a recurrence of a cancer. It's something that's very exciting, and it's happening. It's still at a primitive stage, but this is going to be the future. And these are the PD-1, PD-L1 inhibitors that I was talking about. And this is the personalized cancer vaccine. Okay, now I also mentioned, if you recall, let me see how I'm doing with time. Yeah, I also mentioned that it's inconceivable that patients with monogenic diseases used to suffer with their diseases, and they were declared incurable, even though we know exactly what caused their disease. I mean, just think about it. You go to a programmer, a coder, and you show them a code, and the programmer finds out, I see why your code is not working. There's one extra line here. There's a missing letter here. And they will just fix it, right? And why is this any different from these monogenic diseases? I told you these diseases are caused by a very specific defect in their DNA, something wrong with their code. So why can't we just fix that code? Now, it sounds simple, but it was almost impossible because editing the genome was a nightmare up until, as you all know, the discovery of CRISPR, which really ushered in a completely new era in genome editing. Now, with CRISPR, we can edit genomes at will. And so the future in the treatment of these monogenic diseases will heavily rely on being able to tinker with their DNA and fix the exact defect that caused their disease in the first place. And again, these are landmark papers of how they jumped on this technology immediately to show its promise in treating monogenic diseases, as you see in this paper, for example, or even this paper to treat HIV. And you can, just like you can tinker with the human DNA, you can tinker with the microbial DNA in order to rid it of the antimicrobial resistance potential. And this promises to be a very exciting way of thwarting this pandemic of antibiotic-resistant bugs, superbugs. The last part of my talk will be on common diseases, which I didn't have a chance to talk about much, but I also want to share with you the excitement that's happening in this space. If you were to ask any geneticist about, okay, we understand you guys are really good with monogenic diseases, but you're not very helpful with polygenic diseases. The things that I'm used to as a doctor when I see a patient in my clinic, they have diabetes, they have hypertension, they could have stroke. These are the common diseases I care about. I don't care about your rare monogenic diseases. Now, I could, of course, reply in several ways to this argument, but let's just stick to the idea that, yes, these are more common. Let's say they're more relevant to your practice. What can we do in genetics to make ourselves relevant? And I'm really excited that over the past 7-8 years or so, there's been a revolution in genetic statistics where we're able to finally make use of GWAS. Most of you probably heard of this term, GWAS, genome-wide association study, where you have thousands of patients, thousands of controls, and you try to understand the genetics of the common disease you're interested in. And now, finally, we're able to combine this knowledge into something concrete that's called the polygenic risk score. And just look at the beauty of how this tool and instrument performs. So if you take the distribution of the polygenic risk score in the population, which perfectly matches with what is known about the normal distribution for any disease in the society, and look at those at the highest quintiles of the polygenic risk score versus the lowest, and look at their blood pressure. This is just one example. You can beautifully predict who's gonna end up on the high end of the spectrum and who's gonna end up on the low end of the spectrum, right? And as I said, this is just an example. There's nothing unique about hypertension. Now we're able to do this for diabetes, for asthma, inflammatory bowel disease, you name it. Any common disease you can think of that's been studied on GWAS now has a polygenic risk score, and we're optimizing the polygenic risk score for our local society because, as you know, or may not know, polygenic risk score is heavily influenced by your ancestry. So for the most part, polygenic risk scores have been optimized for Europeans. And so when people try to simply transfer the polygenic risk score to non-Europeans, they noticed a drop in the performance of these scores. And so you really need to optimize it for your local population, and that's what we're doing for a wide range of diseases. Now, it's very important for us to remember here because there are two questions that usually come up when I mention this. A, what's the value or clinical utility? And what am I going to do about it? So obviously, if I'm able to predict something like a common disease from the time of birth, because remember, your polygenic risk score is something you're born with, right? Then I can totally predict who's high risk, who's low risk, and I can stratify my public health system. Instead of being, randomly distributed, it becomes very much focused on who deserves the most attention. That's A. Now, B, is it fatalism? Is it a fatalistic prediction? So if you're born with a high polygenic risk score for whatever disease, does that mean that this is it? There's nothing you can do about it. And I'm really excited about these studies that are showing the exact opposite. So this is, again, in the spirit of showing you landmark papers in this regard. This is a beautiful paper that first looked into this in the New England Journal of Medicine, where they looked at 3 different very large cohorts for the risk of coronary artery disease. And I want you to pay attention to this. In each cohort, they divided patients into low, intermediate, and high risk for their polygenic risk score, right? So they calculate polygenic risk score across these populations, and they determined who's high, who's intermediate, and who's low. Simple enough, right? Now, here's the catch, and here's where things become really interesting. As you know, coronary artery disease is heavily determined by your healthy lifestyle, the diet specifically. So healthy diet, cessation from smoking or abstinence from smoking, not smoking, maintaining a healthy weight, and exercise. So these 4 factors, if you have them all, they consider you very healthy. So that's a very healthy lifestyle. Now, if you have 2 of them, you're considered intermediate. And if you have none of them, meaning you're obese and you have terrible diet and you don't exercise and you smoke, then you're considered really high risk. And look at the beauty of this. Even if you're high risk from a polygenic risk score perspective, look what happens if you maintain a very healthy lifestyle. You slash your risk by 50%. Okay? So just because you're born with a high polygenic risk score doesn't mean that this is the end of the world for you. You can really do something about it. And again, one of the ideas, even though this is still in infancy, one of the ideas is when I tell people that you're really high risk for something, maybe that will motivate you to do something about your lifestyle instead of waiting until something terrible happens to your heart or your brain. And, just another example that's exactly the same with obesity, and you can see the interaction between the polygenic risk score and the environmental factors. So I hope with this overview, I've given you a future where medicine is completely driven by the individualized genomic information of individuals, which can be obtained very inexpensively these days, and will be even cheaper and cheaper as we go forward, as opposed to the status quo where one size fits all, and we just teach one kind of medicine that should be applied to all patients, and the choice is yours. And I hope you've made up your mind that this is the era of genomic medicine, and we can't afford not to jump on the wagon. So with that, I will conclude. Thank you for your attention, and I'll be happy to take questions. Ibrahim?
Yes. So, Doctor, thank you for your amazing talk. Now, the future of 2080 looks really promising. What do you advise us as medical students and future doctors? How can we drive this change or incorporate genes or genomics into our future specialties?
That's an excellent question, and I'm glad you raised it, because I don't want people, and I might have mentioned this earlier in my talk, but I will emphasize it because it's really important. I don't expect this audience to all become, all the medical students in this audience to all become genomicists, right? But I do expect you all to have the bare minimum that will make you comfortable. So when I talk about variants and genome and transcriptome and all different omics, which I guarantee you will be part of your field, whatever it is, omics will chase you wherever you go. So you need to be comfortable with them. And this is your chance. You're medical students. You're here to learn. It's a learning institute. And so you can spend a month, you can spend a summer. Make sure you don't graduate without some minimum knowledge in genomics that makes you comfortable, interacting with these information as they come your way in your field, whatever it is.
Thank you so much.
Mm-hmm.
Thank you so much. I'm gonna just look at it from a different perspective. This is a lot of data, and a lot of individualized data. I mean, who's gonna own that data? We need to plan this, I guess, before 2018. We need to know who's gonna own that data and how would if we're gonna do, let's say, a newborn gene analysis, is the kid is gonna know that all his data is revealed without him knowing, is that something that we need to plan for? Is that something that we need to think of?
Yeah, absolutely. I mean, the ethics of it is a whole new topic, obviously. There are so many ethical aspects that need to be discussed. I mean, you just scratched the surface of it. I will tell you in brief that I think the society will evolve to a point where they fully recognize the limitation of what privacy means for genomic data. I mean, just think about it these days. Your face is no longer your property. Your face is being recorded all the time using face recognition tools, right? And we take it for granted that, okay, this is part of security. And you can see this evolving with other things. Everything you leave this chair, you're leaving traces of DNA that I could analyze. Could you say, I own this chair because I sat on it and it has a piece of my DNA? It's going to become really difficult. And I think the public, as they become a bit more literate in genomics and the power of these tools, they're going to come to recognize that there are red lines we need to draw as a society, and we need to be realistic. And even now, all these big, state-of-the-art data centers, they tell patients there's no way I can guarantee you 100% total privacy of your data, because no matter how much you crypt the data, there's a way to deduce your identity from this data. And it's like the cat and the mouse. It's very interesting to follow this field on data privacy. And every time they're like, okay, we've got it. This time, this is pretty secure. Some, smart hacker would say, nope, I was able to deduce your identity from the data. So I think this is never gonna end. It's gonna continue to be the cat and the mouse until we as a society understand, okay, these are the limits of privacy that we agree on as a society, beyond which we're gonna consider it a criminal act, right? I mean, just think about, okay, one other thing maybe, because I'm really interested in this and it can take me forever, but think of me and my cousin. My cousin owns part of my DNA because we share 1/8 in the case of first cousin, right? So that cousin could basically be the source to identify me. And this is happening already. It's not science fiction. I'm sure you're following the news. It's becoming routine in the US, for example, to tap into Ancestry.com, 23andMe.com. And if I have a third cousin whose DNA is in that database, they will be able to track me down. Now, the society is uncomfortable with this idea that, wait a second, I gave you my DNA to tell me about my hair color, about my predisposition to something, and now you're using it to apprehend a cousin that I've never heard of, right? Again, these are conversations we're gonna have to have, but we need all the stakeholders, not just us, the professionals. We need, representation from important stakeholders as well.
Well, Dr. Fawzan, I have a question. It's a comment as well.
Please.
So thank you so much for this wonderful and very informative presentation. And following your lead of recommending this very important field to students, we need to emphasize the importance of community engagement as well, and how to raise the public awareness about such an important
Exactly.
Initiative or trajectory in the healthcare system. So I would also want to use this opportunity to recommend maybe the creation of a task force of both scientists and geneticists, as well as family physicians or primary care practitioners and other healthcare practitioners, to, along of course with the engagement of the members of the community, to raise an awareness and make all of these dreams from 2080 coming true way earlier. So anything that you would like to add into that? Are there any current initiatives, or are you considering maybe to be a member of a task force or something similar?
Yeah, it's been a challenge, I have to say. First of all, I agree with you 100%, and I couldn't have said it any better. So thank you so much. And we're already seeing the impact of things that are not clear to people. Think of the Healthy Marriage Program. To me, that was a travesty. So it was a very intended effort by the government, very costly, and they want something really good to come out of this. And yet people were just not responding because they had no clue what it is. You asked them, did you get any pre-test counseling? Almost 0% received pre-test. And because the system was not prepared to deal with this, as I said, things have improved. I don't want to be misquoted as being too critical, but yes, in the beginning, things were not planned accordingly. So this is a perfect example where, had we engaged the public into understanding what this entails, until today, every time in clinic I diagnose a patient with a rare disease, they ask me the exact same question. Oh, we've had the Healthy Marriage Program test and we were compatible. So they had no clue what they're being tested for. It's just, one tiny example. So a series of workshops that involve members of the public, a media campaign, so they can relate to this topic instead of being this weird thing. I did work with, there is a science museum being built, and they want to dedicate an entire section to educate the public about DNA. So these are the kind of efforts we really need to have more of in order to have more engagement from the public.